So in this second section of Chapter 11, we're going to focus on the physical methods of control. And so on number 6, object number 6, you'll see that there are 5 major types of physical control methods. And so if you look on the screen, here's your list. So physically, we can control them with heat. We can control them with cold. We can control them with something called desiccation, with radiation, and then finally with filtration. So we're going to work through these 5 physical methods of control in this section. So let's start with the heat, right? So if we look at heat, there's relatively 2 types of heat. So we have moist heat and then we have dry heat. And so with moist heat, what we're looking to do is we're looking to cause coagulation or denaturation. So basically we're looking to affect the proteins with moist heat, cause them to lose their shape, cause them to lose their function, and in turn you can shut down cellular metabolism or shut down chemical reactions. Now the benefit of moist heat is it's short exposures of time. So moist heat happens very quickly and it's very effective. We actually consider moist heat as being more effective than dry heat. Now with dry heat, what we're looking to do is dry them out. We're looking to dehydrate them, cause them to lose water. In turn, we're also trying to alter protein structure here. Main method that we're going to use is like incineration. Now the problem with dry heat is it's going to have to get to much higher temperatures and it's typically going to take longer to do that. So we typically say that dry heat is not as effective as moist heat is. Now, a couple of terms that we want to look at, and you can kind of see some temperatures and some time frames that we're looking at here, but one thing we want to look at are 2 vocabulary words that we look at with microbes. So we have what's called TDT and TDP. So if you look at the first one, TDT, this is thermal death time. So for each microbe, we're given a thermal death time. This is the shortest length of time that it's going to take to kill a microbe at a specific temperature. So when those microbes are shipped to us from lab, remember we told you they tell us the temperatures they prefer, they tell us the pH they prefer, they also will tell us the thermal death time. So basically how long it's going to take to kill them. So the shortest length of time at a specific temperature will kill that microbe. But we also have the thermal death point. Thermal death point is the lowest temperature that's required to kill them. So what does the temperature have to be to actually kill that microbe or kill that sample within about 10 minutes? So definitely some vocabulary words to consider when we're talking about controlling microbes. So if we take a closer look at the heat, let's look at moist for just a moment, and then we'll come back to dry heat. So with moist heat, several different methods we can look at. So the first type of moist heat that's commonly used is steam under pressure. So we add pressure. By adding pressure, we can get the temperature up where we need it to be to actually kill things. This is considered to be a sterilization procedure because the temperature gets hot enough to actually kill everything that's there. A great example of steam under pressure, we think like the pressure cooker, but the lab component of that would be the autoclave. So the autoclave that we have in our prep lab, what it does is it adds extra pressure, 15 psi to be exact. By adding that pressure, it gets the temperature up to 121 degrees Celsius, which is warm enough to kill things if it stays at that temperature and that pressure for 15 minutes. This is really important numbers to remember because anything that goes in the autoclave and stays at those exact measurements, when it comes out, it will be sterile. So this particular type of control method kills everything. What's great about this one, like we just said, is it kills everything. Kind of the downfall of this is, depending on what type of material you put in there, it could damage the material. So you have to make sure that whatever the item is, that it's not heat sensitive, that it's not moisture sensitive, and that the pressure is not going to destroy it. So definitely something that we use. So you can see kind of how the autoclave works. A second method of moist heat is non-pressurized steam. So still going to get it up pretty warm, but because it doesn't add pressure, this one's going to be more time consuming. So you'll see it says that this can actually be from 30 to 60 minutes, whereas remember the autoclave, it only required 15 minutes. The other thing with non-pressurized steam is it's typically a cycle that has to be repeated. So we use this a lot in the canning industry. Sometimes we'll use this with making media in the lab. Now, a third thing we want to mention is, as far as moist heat method, is boiling water. This is something we commonly do in our house. I need you to be aware of the fact that boiling water does not sterilize. It doesn't get hot enough. Boiling water just disinfects. So it kills most of the pathogens, most of the microbes that we're going to see, but not all of them. So boiling, it's going to get obviously 100 degrees Celsius. You've got to leave it there for at least 30 minutes to actually disinfect and kill most of the microbes that are on it. A fourth method of moist heat we see is pasteurization. So pasteurization, obviously something that we use with food. So heat we apply, and it's going to hopefully limit infection, limit spoilage, something that we do what we call the flash method with this. So it doesn't get near as hot, doesn't stay there very long, but hopefully it kills majority of the stuff there. I need you to understand that pasteurized is not sterile. So the milk that we purchase from the store that's pasteurized, it has microbes in it. You think about the benefit of that, I guess, adds to the flavor. So if you put foods and beverages through sterilization procedures, it's going to lose a lot of that flavor. So that's one of the main reasons that we do that. And remember, Louis Pasteur was the scientist that developed pasteurization. So you can see an example of what we use in a dairy farm. So they also mention, so we have moist heat, dry heat, again, also something that we can utilize, not as effective though. Remember, it's a little more time consuming, it's got to get a little hotter. Some main methods of dry heat, one we use in the lab all the time, it's incineration. We turn on that incinerator, it gets really hot on the inside, it ignites, and the purpose is to reduce those microbes to ashes. We can also use hot air ovens or dry ovens in order to kill a lot of the stuff that's there. So very common practices that we'll see. Again, they can sterilize, but again, it's a little more time consuming than you would find the other. Some other terms I mentioned here, we said heat is a factor. Cold can also be a factor. Now the biggest thing with cold is cold's going to be microbistatic, so it's not cidal. Cold's not going to kill stuff. All that cold temperature is going to do is it's going to slow their growth. So if you put something in the refrigerator, if you put something in the freezer, it's just going to preserve them, slow them down, but when they come out and they're able to go to regular room temperature, they're probably going to become more active again. So be mindful that cold is microbistatic. Another method that we use is desiccation. So desiccation is another word for dehydration. So if you can pull the water out of cells, then you can essentially slow down their chemical reactions, because water is a facilitator for chemical reactions. So you slow those reactions down and the microbe becomes ineffective. So definitely something that's used. They also mention lyophilization, which is the freeze drying, that preserving method that we use. Again, sucking all the water out, because if the cells lose water, then obviously they collapse and they die. So quick check for you real fast, and then just a couple more physical methods they mention. Radiation is one that's mentioned here. So with radiation, just to note, this is where energy is emitted from atomic activities. So they mentioned the different types of radiation, ionizing and non-ionizing. You probably aren't familiar with those terms, but familiar with some of the examples under there. So X-rays, UV radiation. We note that radiation can damage nucleic acids. It can damage DNA, can damage protein structure. So it can be a favorable method of control. You actually would be surprised to find out that a lot of the foods you eat have actually been exposed to radiation. It causes them to have a longer shelf life because it kills a lot of the microbes that are on there. So you look at like these raspberries, those were exposed to radiation after 5 days versus those that were not. So you see the fungus that's growing on these versus what's not growing over here. Definitely something that's used also on apples. So a lot of the foods that we see at the grocery store have actually been under radiation to kill the microbes that are on them. Radiation also used in the dental industry, definitely something there. And then the last one I want to mention as far as physical method is filtration. So this is the physical removal, that mechanical removal of microbes by passing either the gas or the liquid through a filter. Now the problem with this is, again, filters are typically for gas or for liquid. So if it's something else, it's a little bit harder to control. And so you have your filter, and as the gas goes through it, it pulls microbes out of the gas, or as the liquid goes through it, it pulls the microbes out of the liquid. The biggest problem we see with filters is viruses. Remember, viruses are ultra microscopic. So they're typically able to go through the filter. So that's a problem that we see with using filtration.